Welcome to the fourth lecture in the series of lectures dealing specifically with the adaptive immune response. More specifically, we are focusing on T cells and T cell responses. What we are going to look at with this last lecture in the series is how antigen processing actually takes place in order for the presentation to occur to the respective T cells, whether it's helper T cells or cytotoxic T cells. With this particular slide, we're going to focus on an overview or a generality of what is happening in the processing pathways. And when I mean processing pathways, what I'm referring to is how is it specifically that the antigen gets associated with MHC in order for presentation to take place to the respective T cell, helper T cell or cytotoxic T cell. There are two general mechanisms or pathways associated with how antigen gets processed in order to present to T cells. In general, we have what's known as the cytosolic pathway, and the cytosolic pathway typically utilizes endogenous antigens. That means antigens that are already within a cell in question. These endogenous antigens are utilized in a pathway including something known as the proteasome complex, where the degradation of the antigen takes place in order to produce peptides. These peptides then are shuttled and eventually associated with class 1 MHC in order for the presentation to take place. The second pathway is the endocytic pathway, and the endocytic pathway focuses on the mechanism of phagocytosis. This is for exogenous antigens, antigens that are on the outside of the cell in question that need to be taken up by endocytosis, more specifically what we focused on for the majority of the lectures, phagocytosis. Again, these exogenous antigens need to be taken up, need to be broken down into peptide components, and then have their association occur with the MHC in order for presentation to take place. For the first pathway, which is known as the cytosolic pathway, I want to take a few steps back and discuss a mechanism that is found within typical eukaryotic cells that is involved in protein processing, that is, the breakdown of proteins. There are proteins that have what are known as having a short half-life, meaning that they don't exist for a very long period of time inside the cell in question. Also, proteins that are abnormal or misfolded need to be degraded because they are not functioning properly. So, there are situations within a normally functioning cell where proteins need to be degraded for one reason or another. So, a normal cellular function and component of that function is something known as the proteasome. The proteasome is a barrel-like structure that is a protease complex. It means that it is a complex that has the ability to digest or degrade or break down proteins within the cell. So again, this is a normal cellular component that we know exists in protein processing pathways. In many situations, proteins that need to be degraded or are targeted for degradation are tagged with another component known as ubiquitin. These proteins that are tagged with ubiquitin typically again mark those proteins for destruction for one reason or another. We're going to take that mechanism one step further and talk about an immune-specific process. There is a pathway used for immune activity that has a distinct proteasome known as the immunoproteasome. The immunoproteasome is induced by cytokines and allows for the degradation of proteins to occur in order to have those peptide fragments that are generated associate with MHC to be utilized in presentation to T cells. So let's look at a little more detail associated with the cytosolic antigen processing pathway. Proteins that are already inside of the cell, those that are endogenous, again, protein antigens that are already inside of the cell. The classic example of this is going to be viral components. Like we've mentioned multiple times, viruses need to enter into the host cell in order for infection to take place. This then involves taking over the cell's replication, transcription, and translation machinery. So this processing pathway would be a great example, or is a specific example, for viral infections. If a virus has already gained entry into the cell, we can then see how this mechanism is utilized in order to process that viral component to be able to present to T cells. So the proteins that might be part of a particular virus enter into the proteasome, specifically the immunoproteasome, and are degraded into peptide fragments. These peptide fragments can be broken down into individual amino acids, or the peptides can be shuttled into the lumen or the space of the rough ER. There is a membrane-associated protein, part of the rough ER, known as TAP, and TAP is short for transporter associated with antigen processing. Its job is to transport these peptide fragments that are produced by the immunoproteasome, transport them into the lumen or the space of the ER. Now, at the same time, the ribosomes that are part of the rough ER, this is why the rough ER gets its name as rough, because the ribosomes are associated with the membrane. The ribosomes are synthesizing the MHC class I protein. That protein is produced and also fed into the lumen or the space of the ER. So within the space of the ER, we now have the class I MHC, along with the peptide fragments that have been generated by the immunoproteasome. There are proteins known as molecular chaperones whose job it is to assist in the assembly of the MHC with the peptide fragments in the space of the lumen of the ER. At this particular point, you have assembly of the antigen with the MHC. This complex then is shuttled to the Golgi via a transport vesicle. Once it enters the Golgi acts as a multifunctional organelle that receives that information, receives those proteins that have the antigen bound to MHC, and then shuttles that off to the exterior surface of the cell, again in a vesicle. The vesicle contains the antigen bound to the MHC. That is then transported to the surface of the cell in order for that presentation of antigen to take place. So let's review that process one more time. You have degradation of the protein component via the immunoproteasome. You then have transport of those peptide fragments into the rough ER via the TAP protein, which is a transport protein part of the rough ER membrane. You then have assembly of class I MHC, which was synthesized by ribosomes on the surface of the rough ER. You have assembly of that class I MHC with the peptide fragment that was just transported in through TAP. The next step is to transport that MHC with the peptide bound, the antigen peptide bound, transport that to the Golgi. The Golgi then targets that complex to the surface of the cell and hence allows for the presentation of that particular antigen. The endocytic pathway requires the uptake of the pathogen via the process of phagocytosis. So in the center of the slide, you see the antigen being taken in. Now this mechanism here is a little different mechanism than what we focused on, but what I want you to do is tap back into your knowledge base of what happens during phagocytosis and focus on the phagocytic mechanism with taking in a potential pathogen and degrading it. Once that degradation has occurred via the phagocytic pathway, what happens is the MHC that has been synthesized by the ribosomes of the rough ER, in this case MHC class II, associates with the components of the phagosome, the phagolysosome, and those peptide components that have been generated by the degradation of phagocytosis are now associated with the MHC that has been produced by the ribosomes of the rough ER. Once you have that association, those components are then sent back to the surface of the cell and allowed for presentation to the respective T cells. Let's review that one more time, as this is also a bit of a complex process. What we have in the endocytic pathway is exogenous antigens need to be taken in. So one of the first things that happens is that you have to have synthesis of class 2 MHC that occurs by ribosomes in or associated with the rough ER. That class 1 MHC is transported to the Golgi and then transported from the Golgi to meet with the contents that have been produced via phagocytosis. So at the same time all of this is happening with the production and the association of class 2 MHC with the vesicle and being transported to the Golgi, we simultaneously have the phagocytic process taking place, with phagocytosis taking in the potential pathogen, degrading it via the process of phagocytosis. The contents then of those broken down products are going to fuse with the vesicle that contains the class 2 MHC so that we can get association with those peptide fragments that were produced by phagocytosis to associate with the MHC class 2. Once that association has been taken place, that vesicle that contains the antigen bound to class 2 MHC is then sent to the surface of the cell in order for presentation to take place. Now, antigens, as I've mentioned previously, are typically macromolecules, and proteins are the number one class or category of macromolecule that acts as a very good antigen or strong immunogen. However, other macromolecules can be utilized as antigens: polysaccharides, lipids, and in some very rare instances, nucleic acids. There is an entirely separate processing pathway for non-peptide antigens, specifically lipid antigens, to be presented to T cells. There is a completely different class of receptors known as CD1 that is the component that is doing the presentation to the T cell. So MHC class 1 and MHC class 2 are specifically involved in presenting peptide antigens, and a separate, completely distinct pathway is utilized for the presentation of lipid antigens, and the molecule that is doing that presentation is known as CD1. Now, it turns out that the processing pathway for lipid antigens is not as well understood as what we've just discussed with peptide antigens. However, there are some similarities in the processing pathway. I should mention that the processing pathway is a complete, distinct, different type of processing pathway, but there are some similarities. CD1 is the molecule that does the presentation, like we've just discussed. It is a protein receptor that is synthesized by ribosomes in the rough ER. It is transported from the rough ER to the Golgi, much like what we just talked about for both class 1 MHC and class 2 MHC. However, this is where there's a little bit of difference in the pathway that takes place, in that self-peptide components as well as pathogen peptide components can in fact be associated with CD1, and CD1 then with the antigen is sent to the surface of the cell for presentation. Now, that's the part that unfortunately there's still a lack in our knowledge. We have a general understanding of how this mechanism works. However, the specifics still need to be worked out. So now let's take another more detailed look at the complex interaction that has to take place in order for antigen presentation, as well as the signal to be received in the respective T cell. So in this figure, we are looking at a blown up version of both the receptors associated with a T cell as well as the cell that is doing the presentation. This is a really nice slide in that it does have things very idealized or stylized, but allows you to see the interaction that needs to take place in order for antigen presentation to occur. As with all of the figures that we've looked at so far, the red dot or circle represents the antigen, and what you see here is the complex association of TCR as well as MHC doing the presentation, and you have the co-receptors CD4 or CD8 that are playing a role in recognizing the MHC, whether it's class 1 or class 2 overall. So just as I've done multiple times throughout the previous lectures, I like to remind all of us that it's not just a simple, straightforward mechanism that we're focusing on. There are, in fact, numerous receptors on both the side of the T cell as well as the cell that is doing the presentation that need to interact in order for the complex recognition to take place between antigen presenting cell, and respective T cell. What you can see from this particular slide is, again, just a snippet of the receptor types that are involved in the interaction between the two cell types that needs to occur. What I do want to focus on, however, is the component that is found in both of these figures, that is a receptor known as CD3. So if we hone in just a little bit and focus right at the center of those figures, where you have the MHC, the antigen, and the TCR interaction taking place, what I've also highlighted in this particular slide is the CD3 receptor. CD3 is an accessory molecule. It is made up of 5 distinct polypeptides, and it participates in the signal transduction mechanism after the interaction with antigen. This particular molecule, CD3, is required for activation. It is required for the signal transduction and the continued activation to take place. Without CD3, the signal is not received, and hence the cell will not behave in a specific way. So this is looking at that interaction in a little more detail, in that you have engagement of MHC with the peptide that's bound to MHC. This initiates a process that leads to the assembly of signaling complex that again allows for signal transduction for the cell to behave in a specific way. So again, to reiterate, the MHC with peptide bound allows for interaction with TCR, and this initiates the assembly of the signaling complex. Once that signaling complex has associated or assembled, the activation of the cell to act in a specific way after receiving that signal can actually take place. With these last 2 slides, what I want to do is focus in a little more detail on the cytotoxic action of cytotoxic T cells. So what exactly are cytotoxic T cells doing? So the first thing I want to reiterate is that the effector cell that is produced by the activation of cytotoxic T cells is a cell known as a CTL. So cytotoxic T lymphocytes are the effector cells. They are the ones that are doing the job of cytotoxicity overall. The very first step in the far left-hand corner on the top of the image, you see the interaction between the TCR on the cytotoxic T lymphocyte interacting with the antigen associated with class 1 MHC in the target cell. So this is that complex interaction that we've been talking about over and over and over again. So that interaction needs to take place. The next step, which if we move right towards the center of the slide, is that you have the cytotoxic T lymphocyte and the target cell form something known as a conjugate. So once that interaction that we've just talked about in the previous image, the one to the left, occurs, that then causes the signal to be sent to allow for this conjugate, which is a very close, tight interaction between the 2 cell types, to take place. Moving on to the right of the slide, we begin to see the Golgi and the granules that are part of the cytotoxic T lymphocyte move towards the site of contact with the target cell. This is in preparation for those components to be released and eventually lead to the degradation of that target cell. So the CTL granule are released or exocytosed, a process that is the reverse of endocytosis. This is allowing for the release of those components to take place. The granule contents are released, and those granule contents then begin the process of apoptosis in the target cell. Apoptosis is also referred to as programmed cell death. It is a highly regulated mechanism, and it is very easily identifiable for those who have been trained in looking at deterioration or degradation of cells. There is what's known as characteristic blebbing, which is these little bubble-like structures that seem to form when apoptosis is induced. There is clear nuclear fragmentation or distinct nuclear fragmentation that takes place, and there is evidence of mRNA decay that also takes place. So there are characteristic physiological things that are occurring in apoptosis, and these are all triggered by the release of the granules from the CTL, targeting that target cell for destruction. So just to reiterate, what makes a target cell the target for destruction by a cytotoxic T lymphocyte? These are cells that are self cells that are altered in some way. They are usually cells that have been infected with the virus, or are cells that are tumor cells growing out of control. There are also examples of cytotoxic T lymphocytes destroying or degrading damaged cells. However, those first two categories seem to be the number one, number two job of a cytotoxic T lymphocyte, is to identify those aberrant cell types. So in this particular figure, what we want to do is look a little more closely at that interaction between the two cell types, the interaction between the cytotoxic T lymphocyte and the target cell. So what we're looking at in step 1, that is labeled as step 1 on this particular figure, is that interaction that occurs between the two cell membranes triggers a rise in intracellular calcium. This rise in intracellular calcium, again, is triggered by the CTL target cell conjugation that actually takes place. This increase in intracellular calcium causes the release of granules, and more specifically the enzymes that are found within those granules, into the space between the two cells. And this is what you're seeing essentially with steps 2 and step 3 in this figure. So what exactly do we know is contained in these granules? Well, there are a number of molecules found within the granules. One of them is a molecule known as perforin, and that's what you see in this figure, those little jelly bean-like structures. Perforin, or perforin, is a molecule that will begin to form polymers in the membrane of the target cell, essentially making holes or pores or openings in the target cell. Once those pores have been formed, the other contents of the granule can now actually enter into the target cell very easily. Other molecules that are found within the granules would be general enzymes known as proteases, which are enzymes that degrade proteins. There are enzymes known as granzymes, which are also proteases that degrade proteins. There is also an enzyme known as fragmentin. Fragmentin is also a type of enzyme that degrades proteins, but it actually degrades a specific subset of proteins known as histones. Histones are proteins that are found in association with DNA. So when you target that particular protein, you begin to see the breakdown of nuclear structure, specifically DNA structure, within the nucleus of the target cell. So in step 4, if we go back to the figure, step 4, you see perforin inserting into the target cell membrane. Step 5 shows those cylindrical pores being formed, and again, this allows for the enzymes within the granules to enter into the target cell and destroy that target cell via apoptosis.